Using this protocol, we can address key questions regarding how microbes assemble and interact in the phyllosphere. It has the added benefit that by using the sterile vegetable extract, we can link the structure of phyllosphere communities to their function during vegetable fermentation. These techniques enable scientists to test how microbes interact in the phyllosphere, and then track how these phyllosphere communities can impact fermentation.
To surface sterilize the cabbage seeds, start by placing 100 seeds in a 1.5 milliliter microcentrifuge tube. Add 1 milliliter of 70% ethanol to the tube and vortex it for 5 minutes. Then use a pipette to discard the ethanol. Add 1 milliliter of 50% bleach and vortex the tube for 5 minutes. Then discard the bleach. Rinse the seeds 4 times by adding 1 milliliter of autoclaved deionized water, vortexing the tube, then discarding the water. After the last rinse, soak the seeds in sterilized deionized water for 2 to 8 hours to soften the seed coat before planting. Weigh 10 grams of clean calcined clay into a 15 by 2.5 centimeter glass tube. Add approximately 9 milliliters of prepared MS broth to each tube to cover the calcined clay. Loosely cap the glass tubes with 22 millimeter two-way test tube caps. Autoclave the tubes at 121 degrees Celsius for 60 minutes. When removing the tubes from the autoclave, push the caps onto the glass tubes to seal them. Then cool the tubes to room temperature before use. When the tubes have cooled, use extra long forceps to gently place 1 sterile cabbage seed into the center of